So far we have known a lot of things regarding immunology. Let's discuss the complement pathways. Complement pathways. First, let's review some things. That the complement is protein, protein in nature. These are proteins of liver, hepatic protein, plasma proteins, and they are a part of our innate immunity. And they have the MAC, which lies there is a screening test CH50, which detect any abnormalities for the classical pathway complement activation. So in complement pathways, I like the UL diagram, and I will explain that diagram here because it's very much simple and easy to understand rather than the first aid diagram. So in complement pathway we have alternate pathway, we have classical, and we have lecithin pathway. So in alternate pathway, the actual triggering factor for activation of complement alternate pathway is the surface of bacteria. Let's suppose this is a bacteria and its surface, it's get attached to C3b, which comes from C3. So C3 gives C3b and C3a, which is an anaphylaxotoxin. So C3b attach to the surface of bacteria, and with the help of factor B, it goes to activate C3 convertase, which is the main pivot for all three pathways. So C3b attaches to the bacterial surfaces, and it's activated by factor B and converts into C3 convertase, and this C3 convertase is then convert C3 to C3p along with C3a, which is anaphylaxotoxin. This is also anaphylaxotoxin. Then C3b is convert C35, C5a and C5b. C5a is also ana and chemotactic agent, and C5b is give rise to MAC, and MAC kills gram negative bacteria cytotoxic with the help of C6 and C9, C6, 7, 8, 9. This is the main stem of the complement pathway. Regarding classical pathway, we have IgM and IgG, which attach to C1 complex, and along with C4 and C2, the C1 convert into C3 convertase. C1, C2, C4, they are converted into C3 convertase, and again the picture remains the same. C3 convertase converts C3 to C3b, which convert C5 to C5b and MAC. And in the lecithin we have mannose or other sugar or sugars. These mannose, they attach to C mannose binding lecithin or C1 like complexes along with C2 and C4. They goes to C3 convertase and which converts C3 to C3b to C5b and this. So far we have learned the three main pathways: C3, C3a, C4a and C5a. They all are anaphylaxis. They take part in anaphylaxis or anaphylaxis toxins. C5a is a chemotactic. It recruits neutrophils. What are the opsonins? Opsonin are the C3b and IgG. Also there are inhibitors for this pathway. So the inhibitor are DAF or CD55 and C1 esterase inhibitor, which prevent autoimmunity, like the complement attack of body's own RBC cells. So the DAF or CD55 and 59 are our C1 esterase inhibitor. These are the inhibitor of complement pathway. C3b also clear immune complexes. So this is all about I guess I have covered all the things which are under discussion. There is a one MCQ over here. As IgG is involved in classical complement pathway, so the most common cause of complement autoimmunity or autoactivation of the complement pathway, classical complement pathway, is autoimmunity against this pathogen. This autoimmunity against this there is immunoglobulins which activate C1 complexes and the pathway goes on. So autoantibodies activating complement pathway. Most common cause of autoimmune classical complement pathway activation is the antibodies which attack C1 and activate them. So this is the, and there is also in macrophages we have no IgM receptor. As IgM is a potent stimulator of C3b, but in macrophages we don't have any receptor for IgM, so IgM indirectly increases or enhance C3b phagocytosis. This is all about complement pathway. Next we will discuss the complement deficiencies.